Welcome to the primer video on the biochemistry of nucleotides like purines and pyrimidines, and also the structure of DNA. Now this is dense and potentially confusing material, but there are some good diagrams in the study guide, and the more you watch this lecture and work on learning these pathways, the easier it's going to become. It's definitely worth your time as you prepare for step one.
It's time for that 3-question warm-up. Let's get going. Which antibiotic fits each of the following drug side effects? First one we have here is teeth discoloration. That's going to be tetracyclines. Next we have tendinitis. Think of fluoroquinolones. Red man syndrome, that's going to be vancomycin. Gray baby syndrome is chloramphenicol. Cartilage damage in children, that's again going to be fluoroquinolones. Side effect of nephrotoxicity, especially with cephalosporins, and ototoxic, especially with loops, that's going to be your aminoglycosides. Remember that the list of drugs that are both nephrotoxic and ototoxic, think of aminoglycosides, vancomycin is another one, but also think about loop diuretics and cisplatin. Next we have pseudomembranous colitis. That can be caused by any antibiotic, but the big ones are clindamycin and ampicillin. Next question, which antibiotics are safe during pregnancy? So basically all the penicillins and the aminopenicillins, also piperacillin and the cephalosporins, most of those you can use. The macrolides, specifically erythromycin and azithromycin are pretty safe. Metronidazole is pretty much safe throughout, but we try to only use this after the first trimester. And then nitrofurantoin can be used, and we're commonly using this for UTI treatment. Next question, what structures form Hasselbalch's triangle? So imagine that triangle kind of pointing down and toward the groin. Now the top of that triangle is the inferior epigastric artery. The medial border is the rectus abdominis muscle. And the lateral border is the inguinal ligament. All right, that's it for the warm-up. Let's get to that lecture.
Welcome to biochemistry. Now, biochem is something a lot of students dread, so we've divided the biochem videos into two groups. The first 6 videos deal with DNA and RNA and genetics, which is a little less intimidating for most students. Then later we're going to come back and do the rest of biochemistry with glucose metabolism and the TCA cycle and the electron transport chain and all that. But first, let's talk about DNA. DNA is really, really long, so in order to pack it all into the nucleus, it hangs out in a super condensed form called chromatin, which is basically DNA and some specialized proteins called histones. 8 specific histone proteins come together, and then the DNA strand wraps around them twice, and that unit is called a nucleosome. So the nucleosome consists of a core made of histones, and then the DNA is wrapped twice around that nucleosome core. And specifically, the core histones are histone 2A, histone 2B, histone 3, and histone 4. And you get 2 of each of those for a total of 8 histones in the core. Now, those histone proteins consist of a lot of lysine and arginine, which makes the histones positively charged. And that positive charge makes it pretty easy for the histones to associate with DNA, which is negatively charged. Now, why is DNA negatively charged? Because of the negatively charged phosphate groups. And because the phosphate groups have this negative charge, the DNA is able to loop around the positively charged histones. So the DNA wraps twice around a core of 8 histones like a thread wrapping around a spool, and then you move down the DNA a little farther and you come to another nucleosome. So it's kind of like beads on a string where the nucleosomes are the beads. And then you have histone H1, which is the only histone that is not in the nucleosome core. And histone H1 is what ties one nucleosome to the next nucleosome. And that whole structure is chromatin, which helps pack a lot of DNA into a relatively small space. Now let's drill down a little deeper and look more closely at the structure of the DNA molecule itself. DNA is made up of 2 strands in a double helix, and the backbones are made up of deoxyribose linked together by phosphate groups. And then between the 2 backbones, like the rungs on a ladder, are the bases, adenine, guanine, cytosine, and thymine. And in RNA, instead of thymine, you have uracil. Now sometimes we call these nucleobases because when you take that base and you add the deoxyribose and a phosphate, then you have a nucleotide. Cytosine, thymine, and uracil are pyrimidines, and adenine and guanine are purines. Now I'm sure you remember that A pairs with either T or U, and C pairs with G. But there are a few picky details that you may have forgotten that are worth remembering for step 1. First, if you take cytosine and you remove this amino group, you get uracil. So deamination of cytosine makes uracil. And uracil is found in RNA while thymine is found in DNA. Another thing, The bond between the guanine and cytosine in complementary DNA strands has 3 hydrogen bonds, but the bond between adenine and thymine on the complementary DNA strands only has 2 hydrogen bonds. So as a result of this, the segments of DNA that have a higher content of C and G are going to have more bonds, and therefore they're going to have a higher melting temperature. So it's going to be more difficult to break those strands apart because of those additional hydrogen bonds. And that's definitely a testable point for step 1. We're going to talk about the synthesis of purines and pyrimidines in just a second, but first let's just briefly mention where the different ingredients come from. To make purines, again there's adenine and guanine, you need the amino acids glycine, glutamine, and aspartate. You don't necessarily need to know where each specific carbon and nitrogen comes from. That was more for undergrad. But it is worth knowing that those 3 amino acids are used in the making of purines: glycine, glutamine, and aspartate. Plus the vitamin tetrahydrofolate. Now, tetrahydrofolate is the metabolically active form of folic acid in your body. And this is one of the reasons why you need folic acid to be able to synthesize DNA. So the carbons in purines come from tetrahydrofolate, glycine, and also from CO2 or bicarbonate. So CO2, glycine, and tetrahydrofolate are the sources of carbon for purine synthesis. So that plus glutamine and aspartate, which contribute nitrogens, makes a total of 5 substances needed to make a purine. And again, you don't need to know which substances contribute which carbons, but I would know those 5 substances are required to make a purine. Then for pyrimidines, you use aspartate and something called carbamyl phosphate. Now carbamyl phosphate basically has 1 carbon and 1 nitrogen. The carbon comes from CO2 or bicarb again, and the nitrogen comes from glutamine. And then the phosphate part of carbamyl phosphate comes from ATP. That phosphate doesn't end up in the finished pyrimidine or the finished nucleotide, but the point is that the synthesis of pyrimidines requires ATP, it requires energy. So the 4 things needed to make pyrimidines are aspartate, glutamine, CO2, and ATP. And those last 3 together are used to make carbamyl phosphate. The enzyme that makes carbamyl phosphate is called carbamyl phosphate synthetase 2, or CPS2. This is a very important enzyme to know for step 1 because it's the rate-limiting step for pyrimidine synthesis. The good news is you don't have to memorize a bunch of biochemical pathways for step 1, but you do need to know the rate-limiting enzymes, plus some enzymes that are deficient in certain diseases, or enzymes that are inhibited by key drugs. I'm going to point those out as we go along. So CPS2 is the rate-limiting enzyme in pyrimidine synthesis. You can probably guess that there's also a carbamyl phosphate synthetase 1. Now, don't get those 2 enzymes mixed up. Let's talk about the differences between CPS1 and CPS2. Take a look at number 4 in your study guide. There are 3 things I want to highlight. First of all, they're involved in different pathways. CPS1 is involved with the urea cycle, which we're going to talk about in a later video. But CPS2 is involved with pyrimidine synthesis. The second difference is the location. CPS1 is found in the mitochondria. CPS2 is found in the cytosol. And then the third point is that the sources of nitrogen are different. CPS1 uses nitrogen from ammonia, and CPS2 uses nitrogen from glutamine. Now let's talk about how we synthesize pyrimidines. Even though purines and pyrimidines are similar in structure, the way our bodies make them is totally different. Even the part of the purine ring that looks just like a pyrimidine is made in a completely different way, which is why we have a different list of ingredients. Fundamentally, when you synthesize pyrimidines, you start with a base called orotic acid, and then you add a sugar to that base. Now, when we look at purine synthesis in a moment, you're going to see that we start with a sugar, and then you add the base. But for now, let's just worry about pyrimidine synthesis. Take a look at number 5 in the study guide. Again, you don't need to memorize the entire pathway, but focus on the key enzymes, which we're going to fill in as we go. First of all, right at the top of this diagram, there's a misprint in your study guide. You're starting out with CO2 and glutamine, so it's glutamine, not glutamate. It's correct on the screen. But again, you start pyrimidine synthesis by converting CO2 and glutamine, plus some energy from ATP, into carbamyl phosphate. And that's the rate-limiting step. And the enzyme that catalyzes this is carbamyl phosphate synthetase 2, and that's enzyme A. Next, you're going to go through several steps to generate orotic acid. And then from there, you can add PRPP, or phosphoribosal pyrophosphate, to make UMP, or uridine monophosphate. So PRPP provides the sugar and the phosphate for both the synthesis of pyrimidines and purines. So again, you started with a base, that orotic acid, then you add PRPP and you get UMP. And enzyme B that catalyzes this is UMP synthase. And I want to point this enzyme out because if you're deficient in UMP synthase, you're going to have a disease called orotic aciduria. We're going to come back and talk about that in a little bit, but for now, just know that enzyme B is UMP synthase. Now, once you have UMP, you can add a phosphate and convert it to UDP. Then you can either convert UDP to CTP, which is cytosine triphosphate, or you can convert it to deoxy-UDP. And enzyme C is ribonucleotide reductase, which converts UDP to deoxy-UDP. That's another enzyme worth knowing, as we're going to see in a moment when we come back to letter D. So if you remove a phosphate from deoxy-UDP, it becomes deoxy-UMP. And there's another important enzyme, which is thymidylate synthase. Thymidylate synthase is enzyme E. We're skipping D for just a moment. So thymidylate synthase converts deoxy-UMP to deoxy-TMP. And this process uses that metabolically active form of folic acid, which we call tetrahydrofolate, or THF. You have to have folic acid in order to synthesize DNA. Thymidylate synthase uses a carbon from tetrahydrofolate, and that leaves you with dihydrofolate. And then in order to regenerate tetrahydrofolate, you need the enzyme dihydrofolate reductase, which is enzyme G. So that's another step worth knowing. Now, the reason these enzymes are important to know is that there are some useful drugs that inhibit these enzymes. So going back up to letter D, the drug that inhibits ribonucleotide reductase is hydroxyurea. Hydroxyurea is used in the treatment of sickle cell disease and is sometimes used to treat cancer. Letter F is 5-fluorouracil, which is another cancer drug that inhibits pyrimidine synthesis by inhibiting thymidylate synthase, which was letter E. So 5-FU is an analog of uracil that irreversibly binds to thymidylate synthase, and it blocks this pathway. We're going to talk about 5-FU more in oncology when we talk about the cancer drugs. And then letters H and I are both drugs that inhibit dihydrofolate reductase. Letter H is the antibiotic trimethoprim, which works in prokaryotes, and then letter I is methotrexate, which works in eukaryotes. Now, we mentioned methotrexate as a DMARD that's used to treat rheumatoid arthritis. It's also sometimes used to treat certain types of cancer and also molar pregnancies. It's a folic acid analog that inhibits dihydrofolate reductase, which means you can't regenerate tetrahydrofolate, and that slows down the production of thymidine, so you can't make DNA. Now, back up on letter B, we mentioned a disease called orotic aciduria, which is caused by a deficiency of the enzyme UMP synthase. So when that UMP synthase doesn't work, you're not able to convert orotic acid to UMP. And that means that orotic acid is going to start building up in the blood, and then it's going to spill into the urine. So it's orotic aciduria. And take note, it's not erotic acid like 50 Shades of DNA. It's orotic acid. But orotic aciduria is an autosomal recessive disorder, and it causes elevated orotic acid in the urine and also megaloblastic anemia. So these patients aren't able to make pyrimidines, and that's a problem for rapidly dividing cells that require a lot of new DNA, such as hematopoietic cells. So this is going to be an unusual cause of megaloblastic anemia. Most common causes of megaloblastic anemia are vitamin B12 deficiency and folate deficiency, because those block that tetrahydrofolate part of the pyrimidine pathway. But with orotic aciduria, the problem is not B12 or folate deficiency. So this is going to cause megaloblastic anemia that cannot be corrected by giving B12 or folic acid supplements. Then a third distinguishing feature of orotic aciduria is that it does not cause hyperammonemia, or high ammonia levels. And that's an important feature because there's another cause of elevated orotic acid in the urine called ornithine transcarbamylase deficiency, which is a disruption of the urea cycle. We're going to talk more about that when we get to the urea cycle in Biochem 14. But patients with ornithine transcarbamylase deficiency have high orotic acid levels and high ammonia levels because you're disrupting the urea cycle. So if you see a patient with orotic acid in the urine, the next question should be, is the ammonia level elevated or is the ammonia level normal? So high orotic acid plus high ammonia is going to be ornithine transcarbamylase deficiency. Then high orotic acid plus normal ammonia is orotic aciduria, due again to that deficiency of UMP synthase. Then the treatment for orotic aciduria is to bypass the pathway by supplementing uridine in the diet so the patient can go ahead and synthesize pyrimidines. Now let's talk about purine synthesis. So I said earlier that when you synthesize pyrimidines, you start with the orotic acid base and then you add the PRPP sugar. But with purine synthesis, we're going to start with the sugar and then add the base. Compared to pyrimidine synthesis, de novo purine synthesis is much less complicated. So we're still on number 5 of the study guide, but now we're focusing on the right side. Purine synthesis starts by converting ribose 5-phosphate to PRPP. Then the rate-limiting step of purine synthesis is catalyzed by enzyme J, which is glutamine PRPP amidotransferase. And there's another cancer drug called 6-mercaptopurine, or 6-MP, that inhibits this enzyme PRPP amidotransferase. So letter K is 6-MP. 6-MP inhibits purine synthesis by inhibiting PRPP amidotransferase. And we're going to talk about 6-MP in oncology, but remember that the immunosuppressant azathioprine is a precursor or a prodrug of 6-MP. Next, we have several steps that require glycine and aspartate and glutamine and tetrahydrofolate. So again, tetrahydrofolate is critical for both purine and pyrimidine synthesis. But ultimately, you're going to form IMP, which is inosine monophosphate. And then that can be converted either to AMP or GMP. Letter L is the enzyme that converts IMP to GMP, and it's called IMP dehydrogenase. Now, if IMP dehydrogenase sounds familiar to you, and I hope it does, it's because there's another immunosuppressant that specifically inhibits this enzyme. Do you remember what that drug is? It's mycophenolate. So that's letter M, mycophenolate. So that's purine synthesis, or de novo purine synthesis. But with purines, you don't always have to make them from scratch, de novo. You can also recycle and salvage purines. So number 6 in your study guide is the purine salvage pathway. And this is clinically relevant because deficiencies in this salvage pathway lead to very specific diseases. So let's look at the pathway first, then we'll look at the diseases. First of all, when you break down nucleosides like GTP, you start by stripping off the phosphate groups one at a time, releasing energy as you go, from GTP to GDP to GMP to plain old guanosine. Then you strip the ribose sugar off the guanosine and you're left with the nucleobase guanine. Now from there, you can either recycle or salvage the guanine to make GMP again, or you can keep breaking it down to uric acid. So let's follow it down to uric acid first. Guanine is converted to xanthine, and then it's converted to uric acid. And the enzyme that does this is xanthine oxidase, which is enzyme A. Now, where have we come across xanthine oxidase before? Back in rheumatology. We treat gout patients with the drugs allopurinol and febuxostat to inhibit xanthine oxidase, to block the production of uric acid in gout patients. And we also talked about how xanthine oxidase is the enzyme responsible for metabolizing azathioprine and 6-MP. And that means that patients who are on allopurinol will be at increased risk of toxicity from either of these two drugs. And if you look at the middle pathway, IMP is broken down to inosine and then to hypoxanthine. And the same enzyme, xanthine oxidase, breaks down hypoxanthine to xanthine. Now, the point of this salvage pathway is not to turn purines into uric acid, but rather to salvage or recycle them. So enzyme B is an enzyme called HGPRT that can recycle hypoxanthine back to inosine, and it can also recycle guanine back to GMP. So HGPRT stands for hypoxanthine guanine phosphoribosal transferase. It's a phosphoribosal transferase. It transfers a phosphate group and a ribosal sugar group onto guanine to make GMP. Now, we're going to come back to this pathway in a moment, but first, let's talk about Lesch-Nyhan syndrome, which occurs in patients who are deficient in HGPRT. So these patients can't recycle or salvage GMP or AMP, so all the purines are going to get converted to uric acid. So one of the findings of Lesch-Nyhan syndrome is gout because of that hyperuricemia. Lesch-Nyhan syndrome also causes some CNS problems like intellectual disability and aggressive behavior and self-mutilation, which is classically lip biting. You can also see movement disorders with Lesch-Nyhan, especially dystonia, which is muscle contractions that kind of contort the trunk or contort the limbs. And then Lesch-Nyhan is also an X-linked disorder, so it's mainly going to be seen in boys. Then how do we treat Lesch-Nyhan syndrome? Well, you can't do anything about a lot of these manifestations, but we can treat the gout and the hyperuricemia by either giving allopurinol or febuxostat, again, to block the formation of uric acid. Then one other disease to mention very briefly is adenosine deaminase deficiency. If we go back to the pathway on number 6, we see that AMP is broken down to adenosine, and the enzyme here is called adenosine deaminase, which converts adenosine to inosine. And if that enzyme sounds familiar, it's because we saw adenosine deaminase back in immunology, because adenosine deaminase deficiency is one of the causes of severe combined immunodeficiency, or SCID. Remember, we said there are a bunch of genetic defects that can cause SCID, but adenosine deaminase deficiency is the most important one you need to know for step 1. So that brings us to the end of session quiz. So here I want you to pause the video, work through those quiz questions, and then we'll go through the answers. All right, first question. What's the rate-limiting enzyme in purine synthesis? We said it's glutamine PRPP amidotransferase. Then what about in pyrimidine synthesis? It was CPS2. Next question. What are the sources of carbon in the synthesis of purines? So we had CO2, glycine, and tetrahydrofolate are the 3 carbon sources. We also said you need aspartate and glutamine, but those provide nitrogen, not carbon. Then what are the sources of carbon in pyrimidine synthesis? Well, the carbons come from aspartate and CO2, and then glutamine is also necessary to provide a nitrogen in carbamylyl phosphate. Next, which medication inhibits each of the following enzymes? So ribonucleotide reductase is inhibited by hydroxyurea. Dihydrofolate reductase is inhibited by trimethoprim in prokaryotes and also by methotrexate in eukaryotes. Thymidylate synthase is inhibited by 5-fluorouracil. IMP dehydrogenase is inhibited by mycophenolate. And finally, PRPP amidotransferase is inhibited by 6-mercaptopurine, or 6-MP. Next, what accounts for the positive charge of histones? It's the prevalence of the amino acids lysine and arginine. And then what accounts for the negative charge of DNA? It's the phosphate groups. Last question. How many adenine residues are found in a molecule of DNA if one strand contains 2,000 adenine, 500 guanine, 1,500 cytosine, and 1,000 thymine? Well, this one looks pretty easy, but there's a little twist to it. One strand of DNA has 2,000 adenine residues, but you have to remember that a DNA molecule is a double helix, so there are 2 strands. And since adenine pairs to thymine, on one strand you have 2,000 adenines, but over here on the complementary strand you can have another 1,000 adenines, which are paired to the 1,000 thymines on the original. So that gives you a total of 3,000 adenine residues. All right, that's it for the end of session quiz, but let's go over these rapid fire facts. First one is a boy with self-mutilating behavior, intellectual disability, and gout. That's Lesch-Nyhan syndrome. Orotic acid in the urine plus elevated serum ammonia. That's ornithine transcarbamylase deficiency, one of the urea cycle disorders. Then orotic acid in the urine and normal serum ammonia is going to be orotic aciduria. And then megaloblastic anemia that does not improve with folate and B12, that's orotic aciduria again. All right, that's it for Biochem 1. I'll see you next time.